you students should come to Friday. First exams on Monday. It's only over 4 chapters.
Yeah, it's only 4 chapters.
Only 4.
Only 4. Well, today we're going to, all the way back to chapter 15. And there is no weekly skills test this week, so nothing for Monday.
No big deal.
I like that.
4 chapters,
She said it wouldn't be a big deal, she said.
Control of glycogen metabolism. Continue. Okay, so we did allosteric control, now we're going to do covalent control. So this is the second type. Alright, so we start with glycogen, phosphorylase. And just a reminder, this is something that's breaking down glycogen. This is inhibited when it is dephosphorylated. It is activated when it is phosphorylated. In general, your A forms and your B forms, the book talks about A forms and B forms, the Sapling Helmer talks about A forms and B forms. Your B form is the inactive form, and the A form is the active form. So what's kind of the opposing enzyme to glycogen phosphorylase? Glycogen
Synthase.
And it is opposing, it is controlled by basically the same kind of phosphorylation pathway that we're going to talk about, and so it's exactly backwards. This one is activated when dephosphorylated. And this activates the A form. And this is inhibited when phosphorylated. That's your D form.
Why do we use inhibited instead of like inactive, or like is there a reason we use?
You could say inactive, but in some cases, if you say inactive, usually that means that it has no activity at all. If you say inhibited, there might be some activity, but not as much as normal. And so actually that brings up a good point. So down here, allosteric activators can upregulate activity of a downregulated D form. There's actually there was a question on the second homework to that effect.
Yeah.
So I mean, these are two separate levels of control: the allosteric control and the phosphorylation control. And so it's better to say inhibited because there might be some basal activity there still. So phosphorylation is controlled by kinase cascade. I'm used to the other group. I keep looking over here expecting to see what I'm writing up on the wall. This is backwards. I don't know why today, but and we're going to start with protein kinase A. So protein kinase A phosphorylase kinase. And with kinases, they get their phosphate group basically from the ATP. And then phosphorylase kinase is going to phosphorylate glycogen phosphorylase. And so then when this is active, we have phosphorylation. Glycogen is broken down. Every good kinase cascade has got a phosphatase to disrupt it. So there's this phosphoprotein phosphatase 1. This is the opposing phosphatase. The kinase cascade. And this can be inhibited by protein kinase A. Phosphorylation by protein kinase A. Or, yeah, anyway. So this gets confused too. Can be inhibited by a phosphorylated inhibitor, which is activated by pKa. We're going to look at the picture. The picture's so much easier to follow. Some other things that you should know. There are some secondary messengers that are of importance. pKa means what?
Protein kinase A.
Sorry?
Is it CN?
Yes. Cyclic AMP. Okay, so now that goes back to chapter 13. Or 14, what was our first chapter?
13.
13? Yeah. Where we talked about the signaling pathway that increases cyclic AMP. needed by pKa to become active. Phosphorylase kinase contains a calmodulin domain. What do calmodulin domains bind? Tells you in the name.
Calcium?
Calcium. So this needs calcium to be active. And we talked about the signaling mechanism for increasing calcium levels inside the cell as well in chapter 13. And then one more thing I want to write down before we go into pictures. Glycogen synthase. This gets phosphorylated by lots of different things. Lots of different kinases. And this includes pKa and other pKa activated kinases. Look for the signature. Book tends to show it's pink, but it's supposed to be like red and green. So the less active versus the more active, the A forms versus B forms. So protein kinase A at the top, phosphorylase kinase, which becomes more active when it's phosphorylated, which phosphorylates glycogen phosphorylase, which becomes more active when phosphorylated, and then that will go down, break down glycogen. And then we have this phosphoprotein phosphatase 1, which can go and remove some of these phosphate groups. So if we remove the phosphate group from the phosphorylase, remove the phosphate group from the phosphorylase kinase, and so that's going to move it into the less active form. Okay. So we need to take a closer look then at how this is regulated, the phosphoprotein phosphatase 1. This enzyme has 2 regulatory sites that can get phosphorylated. Muscle cells, because this is going to differ depending on the tissue type that we're in. So in muscle cells, the phosphatase is active when it's bound to glycogen. And is phosphorylated at site 1. And that is via an insulin stimulated kinase. So this phosphatase is going to go and remove those phosphate groups from my phosphorylase and my phosphorylase kinase. So I'm stopping glycogen breakdown. I want to synthesize glycogen instead. That makes sense with insulin, because insulin is something we use for high blood sugar. So we have a lot of glucose around. We need to store it. This is what that is saying. It is inactive. When it's phosphorylated at site number 2, that's by pKa. And released from glycogen. So this thing just kind of sits right at glycogen when it's working, and it is released, it goes away from it when it's not working. Or if it binds the pKa activated inhibitor, which has got the lovely name of protein phosphatase 1 inhibitor. So again, this is something that we're looking at for muscle cell control. So yellow is my glycogen, and this whole thing is the phosphatase. So there's a glycogen binding subunit, and then there's the actual catalytic subunit for the phosphatase. So what you see is to indicate that that's the phosphatase active unit. And so one choice is I get phosphorylated at site 1, which is going to activate the phosphatase. And then the phosphatase is already recruited to the glycogen. Any glycogen phosphorylases that are trying to break down glycogen get their phosphate groups removed by the phosphatase. And then that's going to prevent them from breaking down the glycogen. If instead I get phosphorylated at this other site by protein kinase A, then what happens is that I get released from the glycogen. And so this is, it says inactive, but it's really inactive because it's no longer recruited to the site where it will be working on these enzymes. So it's kind of inactive by being distant from the glycogen. But then there's this other choice that's not shown here, and that is this binding instead to an inhibitor, which gets activated by pKa. So pKa activates the inhibitor goes and binds the PP1, and then it can no longer act as the phosphatase. Okay. This is different in liver cells. So in liver cells, this is not directly controlled by phosphorylation. Instead, primarily through use of the inhibitor. So this is PP1. and that leads us to kind of the treaty system for overall liver control. Alright, so this is a map worth knowing. And we've seen the first part of it already. This much of it we've already seen. Okay, so again, that's protein kinase A. It is cyclic AMP to be active. If it's active, we'll phosphorylate phosphorylase kinase. Makes it active, phosphorylates glycogen phosphorylase. Makes this active, we start breaking down glycogen. Okay. At the same time, the protein kinase A is doing that, activating glycogen breakdown through the phosphorylase is also inhibiting glycogen synthesis because it either directly phosphorylates the glycogen synthase or phosphorylates other kinases that phosphorylate the glycogen synthase, and when the synthase is phosphorylated, it's inactive. Okay. Then we've got this whole phosphoprotein inhibitor and phosphoprotein phosphatase system going on as well. So the phosphatase works against the protein kinase A. So if it is upregulated, then it's going to go and remove phosphate groups and make these guys less active, and it's going to make the glycogen synthase more active by removing the phosphate group. This thing here can be inhibited in liver cells primarily then by the phosphatase inhibitor. And the phosphatase inhibitor has to be phosphorylated to be active. And if it is, then it'll bind the phosphatase, and the phosphatase becomes inactive. And this guy gets phosphorylated way up here by protein kinase A. Okay?
Hold on.
It's crazy. It is crazy. Signaling cascades, signaling maps are they're complicated, and they involve crosstalk, which is a really hard thing. So especially when you start throwing in other kinases, there's probably umpteen other different pathways that involve some kinase that then comes in and phosphorylates glycogen synthase. So they are complicated. They're complicated to figure out. They're hard to go and figure out which of these are interacting and what is this particular protein being phosphorylated by. And then we find out it's being phosphorylated in 5 different places, and some activate, some inactivate, and it's like 10 different kinases. So it's complicated. Okay, so questions for you. See what you remember from chapter 13. What kind of hormone receptor will lead to activation of protein kinase A? Okay. Well, majority is in the right place. So what does adenylate cyclase do? Makes cyclic AMP. And we use cyclic AMP to activate the protein kinase A. So my adenylate cyclase was a G protein coupled receptor pathway to activate that. Okay, so next one. What kind of hormone receptor will lead to activation of phosphorylase kinase? Does not win on that one. So you have to remember what this pathway does, what the whole point of the phosphatidylinositol pathway was. That was the calcium release. It eventually leads to the ER releasing calcium into the cell. And your phosphorylase kinase has the calmodulin domain and needs calcium to be active. And that was also a G protein coupled receptor. Next one, what kind of hormone receptor will lead to activation of insulin-stimulated kinase? If you haven't figured this this out already. There's only 3 choices. They're the same 3 choices. I just asked you 3 questions.
It's a trap.
There you go there. Yay.
Yay.
Insulin is the tyrosine kinase. And so the tyrosine kinase pathways, we talked about RAS, and we talked about kind of briefly the insulin receptor. Mostly that was in the reading, but it's a tyrosine kinase receptor. All the other things we talked about were G protein coupled receptors. That might be worth writing down somewhere. Oops. Oh, yeah, that's where was that? Back with the secondary messengers of importance. So it's going back a page or so. Secondary messengers of importance, pKa needs cyclic AMP to be active, and this is through a G protein coupled receptor that activates phenolate cyclase. Phosphorylase kinase has commercial domain and needs calcium to be active, and so this is activated through a G protein coupled receptor, in the phosphatidylainositol pathway. Moving forward a little bit, where we mentioned the insulin stimulated kinase. Okay. This is via tyrosine kinase receptor. So when we're out to insulin, we're now talking about hormones. So that's what the last little bit that we need to write out. So hormonal control. Involved in glycogen metabolism. Those include insulin. What was the other peptide hormone? Glucagon. And then there are catecholamines, epinephrine, and norepinephrine, but there's epinephrine. Okay, so these hormones bind cell surface receptors. Stimulate increase or decrease of secondary messengers and to affect kinase cascades. hormonal promotion of glycogen breakdown. And again, this is different depending whether you're talking about liver or muscle, which is our two main places where glycogen gets made and stored. So we're going to start in the liver. Okay, your pancreas releases glucagon in response to low blood sugar. Binds a G protein coupled receptor. Resulting in an increase in the cellular levels of cyclic AMP. And then when cyclic AMP activates PKA, and the resulting kinase cascade. To upregulate glycogen breakdown. Okay, low blood sugar. I need more glucose in my blood, so we're going to break down the glycogen in the liver. That's the liver's. The liver does a lot of things for us, but there's a big one is just kind of maintaining blood glucose levels. Okay. Glucagon does not act alone. We've also got epinephrine and norepinephrine. Epinephrine are released by the adrenal gland. Also in response to low blood sugar. Two different receptors. A beta receptor or an alpha receptor. Okay, these are both still G protein coupled receptors. This one is going to lead to an increase of cyclic AMP levels as well. And the alpha receptor leads to an increase in calcium levels. So cyclic AMP activates PKA again, and our calcium activates the phosphorylase kinase. Or upregulates. So, combination of signals. All lead to breakdown of glycogen in the liver export glucose into the bloodstream. Cartoon version. So right now we're just looking at this side of it, because this is our liver cell. So we've got these different receptors, the glucagon receptor, the beta receptors, and alpha receptors for epinephrine. The glucagon receptor, the beta receptor, will be associated with activation of adenylate cyclase. We make cyclic AMP, we activate AKA, we break down glycogen, that can be transported. The liver cells, they have a glucose transporter to bring glucose into the cells, but it's not really regulated. Your liver is always just bringing glucose into the cell, and it's just based, how much is just based on how much is in the bloodstream at any given time. So that isn't necessarily under any of these hormonal controls. Okay, but now I got the liver cell, or sorry, the muscle cell. So in muscles, epinephrine binds beta receptors to increase our cyclic AMP. And that's really the only biggest thing there. So there's no glucagon receptor on muscle cells. It's something that's just on the liver cell. And we don't have to worry as much about stimulation and calcium release, because in muscles, which is a section we skipped, right? You know, I don't remember what chapter that was, chapter 7, way back last semester. There is a section that talks about muscle contraction, and so it's got a whole different system for releasing calcium, because contraction is tied in deeply with recovery and release of calcium inside the cells. So that isn't controlled under alpha receptors. Those don't exist on the muscle cells. So again, here's my epinephrine, here's my beta receptor. It's going to stimulate cyclic AMP, which will upregulate glycogen degradation. Okay. So now we're ready to do the other one, promoting glycogen synthesis. We're going to do this backwards. We're going to talk about muscles first, because that's actually more complicated. Or other tissues. And in this case, muscle is referring to. Okay, is this the only other major cell type that makes and stores glycogen in large quantities? So, pancreas releases insulin in response to high blood sugar. Insulin binds receptor tyrosine kinases. several kind of downstream effects. Okay, this is going to activate insulin stimulated kinase. What does that go and do? It's going to phosphorylate something. What's it do? Yeah, okay, so we're going to phosphorylate the insulin stimulated, oops, I'm sorry. The phosphoprotein, protein phosphatase at site, what? Site number 1. This is going to lead to a downregulation of PKA activated kinases, or we'll just say enzymes. So this is going to basically inhibit glycogen breakdown. And it's going to lead to an upregulation of glycogen synthase. So we have the opposite effect. There's the glucagon. So back to our pictures. This isn't shown. So insulin binding to an insulin receptor, yay, glycogen synthesis, but I kind of left out all the things. But yeah, we're yanking off all those phosphate groups that activated the PKA pathway, and then we are yanking off the phosphate group that had inhibited the synthase to make it active. And then notice that in the liver, there's no insulin receptor on the liver. Okay. Insulin affects glucose uptake and glycogen synthesis in other tissues. It does not affect it for your liver. So that's different. So maybe your liver, glycogen synthesis. I'm sorry, can I write one more thing after the? We have to have that bullet point here. Is uptake of glucose into the cell. You might not remember this. We did talk about that last semester as well, where we were talking about examples of transporter proteins and those GLUT transporters, which were examples of transporter proteins where you bind glucose on one side, conformational change, release it on the other side of the membrane. All of those, they're held inside the cell in membrane-bound vesicles that then fuse with the surface of the cell through the stimulation of insulin kinase receptors. So, and this is all stuff that we're not going to have time to go through. It's unfortunate, but these receptors, they're always present in the cell, but they get moved to the cell surface whenever insulin is there to trigger the pathway. And that way, not only are you turning on all the enzymes needed to synthesize glycogen, you're also bringing the glucose into the cell. So we're just going to leave it at this, that yes, we get uptake of glucose into the cell when insulin binds to its receptor. Okay, so sorry. Glycogen synthesis in the liver is mostly controlled sterically through concentration of G6P. So it's not there is no really any hormonal control for glycogen synthesis in the liver. If you've got a lot of G6P around, you're going to make glycogen. And that's pretty much the end of it. Now we do have the hormonal control on the other end to prevent glycogen synthesis and instead do the degradation. So that does exist, but the liver synthesis is all based on concentrations of glucose. So these two pictures that I put up here, these are from the old book. And I actually like them better because they're a little simpler to look at. This is the picture in the new book where they tried to make the cells actually look like cells. And then they threw in little pictures. Here's your pancreas, and here's your kidney gland. Which is great, but when I look at this, it looks busier to me, and I have trouble following it. So I will post this so that you have access to the simplified diagram as well. But it says the exact same thing. Everything that's in the slide is either there or implied in the other one as well. Any questions? Okay, so next is gluconeogenesis, and that's the last little bit of glucose metabolism that we have to talk about, and that wraps up everything for glucose for the exam. And that's why there's 4 chapters on this exam, is because they all sort of go together. And then what comes next is all the citric acid cycle, oxidative phosphorylation stuff, and all that sort of goes together. But we're going to stop there because I don't want to open the gluconeogenesis can of worms and how it's regulated, which is another kinase, phosphatase, glucose.